Okay, so my name is Johann, and my favorite ice cream flavor is mint. And I'm a geneticist, and that's the reason why I talk today about genes and genetic background of central nuclear myopathy. And that here is the place where I'm working. That's the IGBMC in Strasbourg. So now you might think, okay, look at the picture. It's in the middle of nowhere. You're not completely wrong. So we are not directly in Strasbourg. We are south of Strasbourg in a small town with a very typical French name, Illkirch-Grafenstaden. And if you have never been to Strasbourg, then I strongly encourage you to come because we have our beautiful cathedral, which was the tallest building in the world for over 140 years, and we have the most beautiful women in Strasbourg. So the IGBMC is a major center for biomedical research in Europe. And if you want to come, you are really invited. We see on the next slides that some of the people that you might know very well came to see us, so it's really great. You can come, but you should be aware of one thing is that in France there is no law protecting children from work. So if you come with your kids, then I will make them work in the lab as we did here, for example, with Michel, and I will not pay them. So in our team, we are working on congenital myopathies. And in contrast to dystrophies, there is no muscle fiber degeneration. So that's a structural thing. That's why we could call them also structural myopathies. And we can see the structural anomalies on the biopsies. So we take the biopsies, we do some chemical staining that we call histology, and depending on what we see, we can distinguish between the 10 to 15 different congenital myopathies. So if we see, for example, a picture like this, on this green, bluish staining, and we see some red aggregation, it's called tubular aggregate myopathy. If we see on this blue staining here, some areas where there's obviously no blue staining, then we call this central core disease. And if on this pink staining we have this abnormal centralization of the nuclei, then this is called central nuclear myopathy. So again, we have 10 to 15 different congenital myopathies. Take the biopsy, and by seeing what is the structural defect, we can distinguish between the different congenital myopathies. And so in our team, we're working on all of these conditions with a specific focus on central nuclear myopathy. But there is not only one central nuclear myopathy, there is different forms of central nuclear myopathy with a different severity and a different age of onset. And this depends very much on the gene and on the mutation. And today I'm here to explain you what a gene and what a mutation is. Let me first tell you one thing, that science is a bit like football, so it's not the World Cup. Today's England is playing, tomorrow France is playing. And science is a bit like this. So we are a team with complementary expertise, and we can only work together. So on the football field, it would be like there's a defense with a goalkeeper that you might recognize. So that's the genetic part of the team. So we are trying to identify the genetic cause of the disease. Once we have identified the genetic cause, we pass the ball to the midfield. And these people here are then trying to understand what really happens in the cell. Why a muscle disorder is developing in the cell, the Petri dish, but also in the animal model. That is cell biology. So they are trying to identify the pathomechanism of the disease and to identify targets for therapeutic approaches. And once they have understood this, then they pass the ball to the strikers, and they will score the goal. So what was important to know is when you have a very good striker, as in our team, our number 9, then sometimes other clubs are also interested in these strikers. So Belinda was transferred to Dynacure, which is a big loss for us, but not for you, because she's continuing what she was doing before, developing a therapy for central nuclear myopathy. So it's important to show this, because what I want to say with this, that with such a team, you will never score a goal and never win a championship if you don't have a good defense. Because identifying the genetic cause is the precondition to be able to understand the disease, which is the condition to be able to develop a therapy. So knowing the gene is really the first step to develop a therapy. And so as I'm a geneticist, I will talk today about genes and mutations. So let's go back a bit to school. So you might remember that we have a couple of chromosomes. Chromosomes are nothing else than compacted DNA. And a mutation or genetic cause is a mutation on one of these chromosomes. So we all have 46 chromosomes. 22 pairs, they are ranked by size. Chromosome 1 is the biggest, the smallest ones are 21 and 22. And then depending on your gender, you have XY or XX. And now we are trying to identify the genetic cause on these chromosomes. And what we do is that we read these chromosomes as we would read a book. And some of you have certainly read some books. So we can compare indeed the DNA with the book. So if we say that the DNA is a book, then we can say also that the chromosomes would correspond to a chapter. Within a chapter, you have some sentences, so this would correspond to a gene. So a gene is nothing else than a sentence within a chapter within a book. And the book is your DNA. And within sentences, we have some words. At the genetic level, we call this codon. And the codons are composed of 4 letters, C, G, A, T. So we really read DNA like a book. It's quite a big book, because you might have read this book here, Harry Potter, especially in England. I guess that everybody has read this here. So quite big book, quite interesting. I counted the characters on the way to London, because I had nothing to do in the train. And I counted 1 million letters in this book. Sounds like pretty big, but it's nothing compared to the DNA. In the DNA, you have 6 billion characters. So this corresponds to 6,000 of these Harry Potter books. So if we now have a patient and we want to read the DNA, actually we are reading the content of the entire Hogwarts library and not of a Harry Potter book. Okay, and this is quite a big work. And a mutation, basically, is nothing else than a misspelling. So there is, in this big library, in one of these books, in one of these chapters, in one of the sentences, in one of the words, there is one misspelling that explains the disease. And this is what we are doing. We are trying to identify the misspelling. So every time we get a patient, we are reading the entire library, and we are trying to identify this misspelling, which is, again, the first step to develop a therapy. So this is quite challenging, as you can imagine. Because first of all, technically, how can we read a library? How do we identify a misspelling? And how do we know that the word is misspelled? Because the language is quite complicated. I show you this on this picture here. This is DNA language. So we have here on this page 3,000 characters. And per patient, we have 2 million of these pages. Okay, I tell you there is a misspelling on this page. Where? If you don't understand the language, quite difficult, right? So what we need is, first of all, that we need a machine that reads the DNA. That is called a sequencer. So when we say that we read the DNA, actually we are sequencing the DNA. That's like reading DNA. So we need a sequencer that is able to read the DNA. And then we need a collaboration with a computer scientist to be able to interpret the misspellings, which is not easy. Here, again, this is 1,000 characters, okay? So this is here the reference genome, so the reference DNA. Then I analyzed myself, and I saw this. So I have here misspelling here. I have an A instead of a G, but what does it mean? So is that the reason why I like mint ice cream? Or will I develop a disease in 20 or 40 years? So that's the interpretation that is quite challenging, because we have a big number of genes. So all of us, we have 20,000 genes, and we all have many, many, many misspellings. And all of us, we carry 700 rare misspellings. So I'm carrying 700 misspellings that most of you do not have, but I don't know what the meaning is. So this makes it quite challenging to identify the genetic cause. So what we have established now, to be able to identify the genetic cause, we have established now a 2-step analysis. This is now established in since one year in France, but also in many other countries now. And this was quite well. Because we realized that we all have 20,000 genes, but not all of these 20,000 genes play a role in muscle. Actually, we are aware of 200 genes playing a role in muscle, because when there is a misspelling in these genes, there is myopathy. So there are 200 known myopathy genes. And these are here the 200 known myopathy genes. Okay? So what we do now is that instead of reading all 20,000 genes, we only read 200. This is 100 times less work, so 100 times easier to understand and to interpret. And so this is here the list of the 200 known genes associated with muscle disorders. Now we have developed a pipeline with a computer scientist. So if we now see a patient and clinically this patient looks like central nuclear myopathy, then we type this in our program and say, okay, this is a patient probably with central nuclear myopathy. And there will be some genes that will pop up. And then we will preferentially, and first of all, look if there are not any misspellings in those genes that are highlighted in red here. BIN1, dynamin2, ZAC, SPEC, ryanodine receptor, titin, MTM1. So these are the first genes that we analyze. And if we don't find anything, no misspelling in these genes, then we go over to analyze the 193 other genes. So this is currently our strategy, and it works quite well because it helped to solve within a short time more than 60% of the cases. More than 60%. So what took years before, and many of you have experienced this, to have the molecular diagnosis, with this approach, for over 60% of the patient, it takes only weeks. So there is a patient with a muscle weakness. We take a drop of blood, we extract the DNA, we analyze the 200 genes, and then for many of them, for over half of them, within a couple of weeks, we have the result. And having the result is the first step to go towards a therapy. So this is quite good. Over 60% sounds good, but then you might think, okay, then there must be some 40% or so that don't have a misspelling in one of the known genes. Yeah, that's exactly the issue. So this is very helpful for over 60% of the patients, but still we have almost 40% of the patients that do not have a misspelling in the known genes, which means that there is probably new genes to be identified. And this is the second step of the analysis. First step is the panel sequencing. Many of you have gone through panel sequencing, 200 genes. If we don't find anything, then we go over to analyze all genes, 20,000 genes, 100 times more work, so 100 times more difficult to interpret. And this is called exome sequencing. So first step, panel sequencing, only a selection of genes, and then exome sequencing for those we didn't identify the misspelling, and this is all genes. And so we had a pretty big project, the biggest in France and one of the biggest in Europe, where we collected 1,000 DNAs of patients and families with muscle disorders, and then we did this exome sequencing on this 1,000 samples. So this project is called MyCapture, and this is here the result. So in 15 or 16% of the patients, we found the gene and confirmed the gene and gave the diagnosis to the patients. In 22%, we are about to confirm this, so we'll give the result very soon to the patients. In 14%, we have a pretty good idea what the genetic cause is, but look at this here. In 50% of the patients, we don't know what the genetic cause is. And there are three main reasons for this. So the first reason is the approach itself, because exome sequencing is supposed to be analyzed, to analyze all 20,000 genes, but here and then we miss a part of the gene because of the approach. So we might miss, with a low probability, we might miss the misspelling that is causing the disease because of this approach. The second reason is that it is estimated that 20% of all genetic disorders are not due to misspelling in the genes, but due to misspellings in the regulatory regions that control these genes. And this is, we are also doing these things, it's much more complicated to analyze and to interpret this. But the main reason, and this concerns all of you, is that in most cases we do not have enough family members because our success rate clearly correlates with the number of family members that we analyzed. If we analyze only the patient, then we have a success rate of 25%. If we analyze the patient plus the parents, even though both parents are healthy, they serve as negative controls. Then we increase the success rate to 50%. And if we have the patient plus the parents plus somebody else, then we increase our success rate to 75%. So this is the reason why, in your case, if you do not have a molecular diagnosis for yourself or for your kid, that we need the DNA of the parents and of as many family members as we can get, because this is really helping us a lot to analyze the DNA and to filter all these 700 rare misspellings and to clearly identify the genetic cause of the disease. So now I'm coming already to the end. I'm right on time in contrast to Francisco. Well, yes, as always. So one of the major questions now that you might have is, do we now still need a biopsy? Because a biopsy is invasive, a biopsy is painful. Nobody wants to have a biopsy. Does genetic now does everything, or do we still need a biopsy? So if I would ask this question now to my colleagues, I guess that 50% would say, yeah, we need a biopsy. 50% would say, no, we don't need a biopsy anymore. Genetics is doing everything. So I have a very clear answer to this. And my very clear answer is, it depends. It depends on the cases. Now imagine we have one patient with central nuclear myopathy, clinically. So muscle weakness looks like central nuclear myopathy. We take a drop of blood, we extract the DNA, we do panel sequencing. Result within 3 weeks. And now we find a mutation in MTM1. So this fits because it looks like central nuclear myopathy, so it fits clinically and genetically. Then for us, the case is solved. Then we don't need the biopsy anymore. But in the other case, where we have a patient that clinically looks like centronuclear myopathy, we do panel sequencing. We don't identify any misspelling in the CNM genes. We don't identify any misspelling in the known myopathy genes. We go over to the second step. We do exome sequencing. We identify a misspelling in a new gene. Then we need a complete characterization of the disease. Then we need the biopsy, because we really need to understand on the global thing, so what is really going on? And this is super helpful, because if we understand the disease, this will identify novel targets for therapeutic approaches. So here the biopsy does not only help the patient himself, but might help also the community of the patients with centronuclear myopathy. Well, that's all, I think. Thank you very much for your attention, and if you have any questions, I'm ready.